prokaryotic cell, the endosymbiosis to the eukaryotic cell, to an ancient eukaryotic cell. And so we have a double membrane organelle like peroxisome, like mitochondria and chloroplast. So these organelles have their own genome. And they have, for example, for themselves, replication, transcription, and translation, and they made some necessary proteins for themselves. Okay. And so you see in this picture the nucleus, endoplasmic reticulum, and some kind of organelles like Golgi apparatus originate from ER, lysosome originate from Golgi apparatus, and endosome originates from Golgi apparatus, and the organelles of eukaryotic cells originate from Okay, and now we want to talk about the organelles and protein movement between the different part of the cell. You see in this picture, cytosol and different part of the cell, for example, nucleus, peroxisome, plastis, mitochondria, endoplasmic reticulum, Golgi, late endosome, secretory vesicles, lysosome, early endosome, and cell surface. We have some kind of protein sorting or protein transport from cytosol to endoplasmic reticulum, cytosol to plastid, cytosol to peroxisome, cytosol to nucleus, and cytosol to mitochondria. And from endoplasmic reticulum to Golgi, and Golgi to late endosome, secretory vesicles, cell surface, and from late endosome to early endosome, early endosome to late endosome, and late endosome to lysosome. We have three kinds of transport mechanisms: gated transport by gated channels. We talk about in cell membranes. We have transmembrane transport with protein translocators, for example, carriers or pump. And we have vesicular transport, membrane enclosed intermediates, something enclosed in a vesicles and transport between the different part of the cell, for example, between endoplasmic reticulum to Golgi, or from Golgi to cell surface. And now we want to talk about the nucleus and protein transport between cytosol to nucleus. You see in this picture vesicle transport. We have a budding of a vesicle which contains, for example, a glycoprotein, carbohydrate, lipid in the vesicles, and vesicle budding, and then fission to the another membrane in the cell, for example, to the another suckers or another organelles in the cell. Okay. When we want to transport some proteins into different part of the cell, we need a signal sequence, a sequence of amino acid in some part of proteins, for example, in N-terminal, C-terminal, or in middle of the proteins. You see in this picture the signal sequence in N-terminal or N-terminus. You see the signal sequence in this example in the middle of the proteins with red color. So the signal sequence recognized by a receptor, and then the protein transport to the special distinct. Okay, now I said we want to talk about the transport between cytosol to nucleus, and we want to talk about the nucleus. Okay, you see in this picture nucleus. Nucleus have some kind, have some part, and we have a nuclear envelope. The nuclear envelope have three parts. We have outer membrane. You see in this picture, we have outer membrane, outer nuclear membrane. We have inner nuclear membrane, and we have nuclear pore complex, or we said NPC, nuclear pore complex, or NPC. So we have three parts: outer membrane, inner membrane, and nuclear pore complex, and all of them named nuclear envelope. Nuclear envelope. And protein transport from cytosol to nucleus through the nuclear pore complex. Okay. And after nuclear envelope, we have chromosomes or chromatin. Do you know what's the difference between chromosome and chromatin? Do you know what's the difference between chromosome and chromatin? Can you tell me? What's the difference between chromosome and chromatin? You don't know? How about others? What's the difference between chromosomes and chromatin? When we see chromosomes and when we see chromatin? When do we see chromatin and when do we see? Okay, yes. And when we see chromosome and when we see chromatin, when do we see chromatin? Yes, it's correct. Miss Noor, I think. I'm not Frank.
لا مو تسأل لا جاوب.
Chromosome contains DNA and also chromatin contains DNA. Okay, you didn't answer me. We see chromatin in the interface, and we see chromosome in the meiosis, in the mitosis, sorry, in the mitosis. And chromosome is shorter and more thicker, more condensed than chromatin. Both of them are DNA and protein, but chromosome is more thick and short, and it's made of two chromatid. It's made of two chromative mitosis. Yes, we see chromosome in mitosis and chromatin in interphase. Do you understand? We see chromatin. Chromatin is longer, thinner, and less condensed. Yes, yes. But I ask you, what's the difference between chromosome and chromatin? Both of them are DNA plus protein, and the main protein is histones. We have five kind of histones in chromosome and chromatin. But chromosome are more condensed, are thickness are more thick and shorter than chromatin, and we see in mitosis. But we see chromatin in interphase is longer, thinner, and less condensed. Okay, we have nuclear matrix in nucleus. In a nuclear matrix, we have a fibrillar network or laminar network. I'll talk about later. We have a nucleoli, a site of rRNA and ribosome formation, and we have nucleoplasm. Is similar to cytoplasm, but it have a more acidic pH. For example, we have a pH 7 in cytoplasm and 6 to 6.5 in nucleoplasm because of the nucleic acid. So nucleoplasm is similar to cytoplasm. It consists of proteins, lipids, carbohydrates, and some kind of enzymes. But the pH is more acidic than cytoplasm. Nucleoli is a site of ribosome formation and rRNA transcription. We see the rRNA transcription in nucleoli, and we have nuclear matrix. Is similar to cytoskeleton. It's made of intermediate filament. And also we see in nucleus, chromosomes, chromatin, and nuclear envelope is a different part of or component of the nucleus. Okay. If you have any question, please ask me. Do you have any question? Okay. I'm coming. Sorry, I'm coming in a few minutes. I want to answer a phone. Sorry.
Now unmuted.
Okay. If you didn't understand, please ask me. Miss Noor. Right.
ايه جيبي هنا غدا.
Okay. What about the nuclear envelope? We have two cellular membranes. It's a barrier to ions, solutes, and macromolecules. Membrane fused to form pores and complex assemblies of proteins
شنفتين.
named nuclear pore complex, ER. It showed the
خرة تاكل من الصباح.
connectivity of RER, and both of them have ribosomes. The surface of the membrane, of the outer membrane and membrane of RER. An inner membrane bound to nuclear lamina. Nuclear lamina is a skeleton of nuclear. Is a filamentous network, and nuclear lamina supports envelope, shape the nucleus, attachment of chromatin about the inner membrane, and attachment to nuclear lamina. And the nuclear lamina is made of a kind of protein, one kind of protein named lamins. And lamins is an intermediate filament of cytoplasm, and it has three subunits: lamin A, lamin B, and lamin C. It's a superfamily proteins. It has three subunits, at least three subunits: lamin A, lamin B, and lamin C. Okay, you see in this part of the nucleus, we see outer membrane, inner. You see close to the inner membrane and the lamina fibrillar. You see the chromatin, nucleus or nucleoli, and nucleoplasm, the part of the nucleus. Okay, and we want to talk about the NPC or nuclear pore complex. You see the electron microscope graph of NPC, and you see in this picture the schematic figure of NPC. In this picture, you see the different part of the NPC. Okay, we have a cytoplasmic ring in outer membrane. We have cytoplasmic ring in outer membrane. We have cytoplasmic ring, we have nucleoplasmic ring in inner membrane, cytoplasmic ring and nucleoplasmic ring in inner membrane. We have central channel. We have central channel with orange color. We have cytoplasmic filaments, or we see FG repeat or FG filament. It has a repeat of two kinds of amino acid, phenylalanine and glycine. Is a binding site for import receptors. It recognizes the signal sequence, for example, and bend, cytoplasmic bend, and the proteins comes to the pore. And we have a Filaments comes from nuclear ring or nucleoplasmic ring, and we have a nuclear basket in the nucleus. We have a nuclear basket in the nucleus. So we have cytoplasmic ring, we have nucleoplasmic ring, we have central channel, we have cytoplasmic filaments made of two major amino acids, phenylalanine and glycine. We have a nucleoplasmic filament that made in the nucleus a nuclear basket, a nuclear basket. And we have inner ring and outer ring, inner ring and outer ring. Surround the central channel, surround the central channel. Inner ring protein and outer ring protein. Inner ring and outer ring. It's a different part of a NPC. It's a very special structure to transport the very big size molecules. For example, do we have translation in the nucleus or in the cytoplasm? Do we have translation in the nucleus or in the cytoplasm? Do you hear me? We have translation in nucleus or in cytoplasm. Do you hear me? I ask you a question. Do we have translation in nucleus or in cytoplasm? Cytoplasm, excellent, very good. We have translation from mRNA to proteins by ribosomes and tRNA in cytoplasm. So all of the protein make in cytoplasm, and we have a lot of protein in nucleus. A lot of enzymes, for example, DNA polymerase, RNA polymerase, helicase, primase, and some kind of enzyme in the nucleus. And this enzyme should transport from cytoplasm to nucleus because we didn't have or we don't have any translation in nucleus. Yes, do you understand? And you see the importance of NPC. So NPC is a very special structure to transport a very big molecules, very large molecules to the nucleus, very large proteins, enzymes to the nucleus. And we have transcription from DNA to mRNA or tRNA or rRNA only in nucleus. We have transcription only in nucleus. So we have rRNA, tRNA, and mRNA in nucleus. And they should go to the cytoplasm. And they should go through the NPC. So we transport the RNA, some kind of RNA from nucleus to cytosol, and proteins from cytosol to nucleus. Okay, do you understand?
شنو هذا بس دي؟ ها؟
And in this picture, you see the NPC again, nuclear pore complex, and in this picture, and you see in this picture the size of nuclear pore complex, and very large proteins can transport through this pore. But the transport is very especially. Okay. I want to talk about the mechanism of transport. Please listen me carefully. I want to talk about the mechanism of transport. When a protein wants to transport or wants to import to the nucleus, they need a NLS or nuclear localization signal, nuclear localization signal, or nuclear import signal. We said NLS, NLS, or NIS. So the protein wants to import to the nucleus, they need a signal sequence. The signal sequence named NLS or NIS, nuclear localization signal or nuclear import signal. Nuclear localization signal, NLS, direct nuclear protein to the nucleus. We have two kinds of proteins, importing for import the protein from cytosol to nucleus, and we have exporting from nucleus to cytoplasm. So we have importing and exporting, importing cytoplasm to nucleus and exporting nucleus to cytoplasm. Importing is a type of karyophorine that transports protein molecules into the nucleus by binding to a specific recognition sequence called nuclear localization sequence or NLS. Importin has two subunits, importin alpha and importin beta. So importin has two subunits, importin alpha and importin beta. Members of the importin beta family can bind and transport cargo by themselves or can form heterodimers with importin alpha as part of a heterodimer. Importin beta mediate interaction with the pore complex, while importin alpha act as an adapter protein to bind the nuclear localization signal dissociated after binding to RAN GTP inside the nucleus, with the two importin proteins being recycled to the cytoplasm for future use. So we see importin has two subunits, importin alpha and importin beta. Importin beta is necessary for transport for interaction with NPC or nuclear pore complex, and importin alpha is necessary for bind to NLS and recognize the NLS or NIS. So importin alpha know the and recognize the NLS or NIS, and importin beta interaction or interact with the pore complex or NPC and transport the protein to the nucleus. I show you in a picture. In this picture, you see the importing, and you see the protein with NLS, and you see importing. Importing has two subunits, alpha and beta. Importing with subunits alpha bind to the protein, bind to the NLS of protein, and they transport to the NPC. And if you remember, we have cytoplasmic filaments or FG filaments. FG filaments recognize this complex, bend, and this complex go through the NPC from central channel. Importing alpha and protein go through the nucleus, and importing beta come back from NPC. And it doesn't enter to the nucleus. It just enter to the NPC. And after that, come back to the cytoplasm. And transport in a high concentration of RAN GTP is necessary. When we have high concentration of RAN GTP, RAN is a kind of protein that we said GTP binding protein. GTP binding protein bind to the GTP. We have RAN GTP and RAN GDP. The active form is RAN GTP and inactive form is RAN GDP. Importing alpha, beta, and protein go to the or transport to the NPC in a high concentration of RAN GTP. Importing alpha subunit transport by an exportin and come back to the cytoplasm again. Let's see. Receptor by nuclear localization. I said that. Okay, and we have RAN GTP active form, RAN GDP inactive form, and high concentration of RAN GTP in nucleus and low concentration in cytoplasm. You see the mechanism of enter to the from cytosol to nucleus, or import from cytosol to nucleus. We have an importin. Importin has two subunits, alpha and beta. Alpha recognize the NLS or signal sequence, and importin beta interact with the NPC. And we have cytoplasmic filaments recognize this complex, bend, and the complex goes through the NPC in a high concentration of RAN GTP. And then importing beta come back to the cytoplasm, and importing alpha and protein import to the nucleus, and then importing alpha come back to the cytoplasm with exporting or with exit mechanism. I want to talk about the exit mechanism. In exit mechanism, we need NES. We need NES or nuclear export signal. Nuclear export signal, NES or nuclear export signal. And we have a exportin. Exportin is a protein recognize NES. And we need RAN GTP. I'll explain in a picture next slide. So we need NES or nuclear export signal. We need exportin, recognize NES, and we need RAN GTP, okay, that stabilize the complex. You see in this picture, we have a protein with NES or nuclear export signal. Exportin recognize this signal and bind to the protein. And we have RAN GTP. RAN bind to the GTP with 3-phosphate group, RAN GTP. And we have this complex. RAN GTP stabilize the complex of exporting protein. And this complex comes through the export to the NPC and exit from nucleus to cytosol. In cytosol or cytoplasm, we have RAN gap. It's a kind of enzyme that change RAN GTP to RAN GDP. RAN GTP to RAN GDP. Because of this change, the complex or the protein is released. Okay. So we have a protein with NES. We have exportin bind to the protein. We have RAN GTP stabilize this complex. This complex export from NPC from nucleus to cytosol. In cytosol, we have RAN gap. Change the RAN GTP to RAN GDP, and the protein is released. Okay, do you understand? Do you understand? If you have any question, please ask me. You confuse, Mr. Muhammad? You mean confuse, I think. بله سوالات به شکل گزینه هاست. منظورتون تستیه یا چند گزینه ایه؟ بله امتحان به صورت چند گزینه ایه. If you have any question, please ask. Sorry, we have. Close to cytosol. Or what do you know? What do you want exactly? Okay. You see in this picture, run GDP and run GDP. We have an enzyme named RAN GEF. May change RAN GDP to RAN GTP. GF or guanine exchange factor. Guanine exchange factor exchange the RAN GDP to RAN GDP from cytosol to nucleus. And we have RAN gap. RAN gap change the RAN GDP to RAN GDP. Okay? GTP is activating protein. And we have RAN is a kind of GTPase or GTP binding protein. Okay. And I said about the transcription of RNA in nucleus. So we have RNA in nucleus, mRNA, tRNA, and rRNA. rRNA and tRNA goes through the exit mechanism by exporting. For example, rRNA with proteins made the subunits of ribosomes, and the proteins of subunit of ribosome have NES and bind to the exportin and exit from nucleus by exporting. And about the tRNA, they made a complex with proteins, and the proteins have NES and exit from nucleus. But in mRNA, we have another exit mechanism, different from exporting system or exporting mechanism. For mRNA, we have three proteins, DBP5, TAP, and NXT1. DBP5, TAP, and NXT1. They export to the NPC. DBP5 bind to this FG filament or cytoplasmic filament, and they hydrolyze the ATP to ADP, and then TBP and NXT1 release the mRNA and come back to the nucleus. So we have mRNA in cytoplasm, bind to the ribosome, and translation is begin. So for mRNA, we have different proteins to exit the mRNA from nucleus to cytoplasm. DBP5, TAP, and NXT1 bind to the mRNA. And in mRNA, we didn't have NES. We have poly A in cell. We have poly A tail. For example, 100 to 250 adenine, 100 to and now, if you have any question, please ask. the mechanism of transport, import, and export is finished. I want to talk about the laminar network. Do you have any question? Okay. The nucleus envelope is disassembled during mitosis. Okay, the nucleus envelope is disassembled during mitosis. You know why? We have a nuclear lamina in a nucleus is a network of intermediate filament, maintenance of nuclear shape, a special organization of nuclear pores, regulation of transcription, and coring of interphosphochromatin, DNA replication. And you see in this picture the nuclear lamina with electron microscope. You see in this picture what happened when the mitosis, in mitosis, the nuclear envelope is disappeared. We have a network lamina close to the inner membrane. In early prophase, we have 4 stages in mitosis. We have prophase, metaphase, anaphase, and telophase. In prophase, the phosphorylation of lamin is occurred. Phosphorylation of lamins is occurs. Because of these phosphorylations, the lamins network is degraded, and so the nuclear envelope is degraded too. Because of the degradation of laminar network, the nuclear envelope is degraded in prophase, and so the nuclear envelope is disappears. And in telophase, in the end of the mitosis, the phosphorylation of lamin is occurred. We have a phosphatase in this stage, and the phosphorylation of lamin is occurred, and so we have a laminar network, and then the nuclear envelope is formed again. Yes. So phosphorylation, and the phosphorylation of lamin cause of disappear and formation of nuclear envelope in mitosis, in prophase and telophase. Okay. We have some kind of cyclin-dependent kinase or CDK that phosphorylate the lamins, in prophase. And we have phosphatase enzyme in telophase that dephosphorylate the lamins, and laminar network is formed again. Okay. When a nucleus disassembled during mitosis, the nuclear lamina depolymerized. The disassembly is at least partly a consequence of direct phosphorylation of the nuclear lamins by cyclin-dependent protein kinase or CDK that is activated at the onset of mitosis. At the same time, proteins of inner nuclear membrane are phosphorylated, and the NPC disassembled and disappears in the cytosol. During these processes, some NPC proteins become bound to nuclear import receptors, which play an important part in a reassembly of NPC at the end of mitosis. Nuclear envelope membrane proteins no longer tether to the pore complex lamina or chromatin, disperse throughout the ER membrane. The dynein motor protein, which moves along macrotubules, actively participate in tearing the nuclear envelope of the chromatin. Together, these processes break down the barriers that normally separate the nucleus and cytosol and the nuclear proteins that are not bound to membranes or chromosomes intermix. Now, I want to talk about the mitochondria. You see in this picture the structure of mitochondria. Mitochondria is a double membrane organelle. We have outer membrane, we have inner membrane, we have crystal, we have a space between inner and outer membrane. This space or intermembrane space, we said, consists of lipid, water, ions, proteins, and something transport between cytosol and mitochondria may be seen in a intermembrane space. And the inner and the internal space, we said matrix. Internal space, we said matrix. In matrix, we have DNA or empty DNA, the DNA of mitochondria, we said empty DNA. And we have ribosomes. So we have transcription and translation in a matrix of mitochondria. We have some kind of enzyme in the matrix of mitochondria. We have some kind of enzyme in a matrix of mitochondria. For example, we have Krebs cycle, beta oxidation enzymes in the matrix for fatty acids and for amino acids. And we have many kind of enzymes in the matrix of mitochondria. And we have granules or particles in a crystal. These particles named ATP synthase particles or oxysome. Oxysome is used to produce ATP. It's a part of mitochondria to produce ATP. Okay. Do you have any question about mitochondria structure?
Yep, teacher. How are you, teacher?
Okay. And so translocation into the mitochondrial matrix depends on a signal sequence and protein translocators. Proteins, once to transport from cytosol to mitochondria, need a signal sequence. The amphipathic alpha helix, we need some positive charged amino acid and non-polar amino acid as a signal sequence for these proteins. And the structure is alpha helix, amphipathic alpha helix. And we need some kind of protein named translocon or translocation channel, also we said. The translocon from outer membrane named TOM complex and translocon from inner membrane named TIM complex. So we have TOM complex in outer membrane and TIM complex in inner membrane of mitochondria. So we need a signal sequence and two kinds of protein Tom complex and Tim complex in outer membrane and inner membrane. You see in this picture the pathway of the imported protein to the mitochondria. You see the proteins. We see the signal sequence in N-terminus. It's a signal sequence in N-terminus. And you see the proteins with orange color. It's a kind of chaperone. Do you know what's a chaperone? It's a kind of chaperone. No. Do you know the chaperones? What is it, Ms. Noor? Can you tell me? Can you tell me? Chaperone is a kind of protein, especially for folding proteins, and make a three-dimensional structure of proteins. This protein named HSC70, HSC70 or HSP70, HSC70 or HSP70, bind to the proteins and keep protein unfold. Keep protein unfold. Okay. And the signal sequence is recognized with Tom 20 and 22. This number is molecular weight. 20 and 22. It's a molecular weight. So the signal sequence is recognized by Tom 20 and 22 as a receptor. And beside the Tom 20 and 22, we have Tom 40 as a channel protein or translocon protein. And protein transport through the Tom 40. So Tom 20 and 22 recognize the signal sequence. Tom 40 is a channel protein. HSC70 keep protein unfold. And in inner membrane, we have Tim 44. It's another channel protein, is a complex. Tim 44 and Tim 2317. It's a complex or a channel protein, and you see in this picture, outer membrane and inner membrane are close together, or we said contact site. And then proteins transport to the matrix, and in the matrix, the signal sequence is cleaved. The signal sequence is cleaved. And the HSC70 removes the protein, and protein, we see the fold protein that make a active protein in this condition. Okay, I repeat it again. In this picture, we want to talk about the transport protein from cytosol to mitochondria. Okay. In first step, we have HSC17 is a kind of chaperone protein. Keep protein unfold. Okay, in a linear linear condition. And then we have signal sequence in N-terminus. Signal sequence bind to the Tom 20 and 22 as a receptor. And then we have a TOM40 as a channel protein, and we have a contact site. Outer membrane and inner membrane are close together. And in inner membrane, we have TIM44 and TIM23 and 17, and protein import to the matrix. In matrix, cleave the signal sequence, and HSC70 remove the protein, and so we have a fold protein. We have a folding in protein. It's an active form of protein. Okay, do you understand? Yes, it's important. If you have any question, please ask me. I can understand, but what do you want to say? For change in forming gas transport? Just. Yes. Just transport, yes. Just transport. No, it's just transport. It doesn't change in forming. No. New. In previous lecture, we talk about nucleus, and in this part, we talk about mitochondria. Okay. So the form of protein doesn't change. Only transport. You see, chaperone protein is a family protein, and we have a HSP or HSC70 is a member of this family. Interact protein help to prevent aggregation before engaging with TOM complex in outer mitochondrial membrane. And proteins are imported as unfolded polypeptide chains because of chaperone. And we have a contact site that join the inner and outer membrane together or close together. And you see in this picture, again, import the protein. We have a signal sequence protein bind to the receptor or TOM20 and 22. We have a channel, go through the TIM23, TIM17, and TIM44. And in the matrix, clip the signal sequence. And we have a major protein in folding form. Okay. And in this part, I want to talk about the electron movement chain. Electron movement chain. We have 4 complexes in inner membrane in crystal part. We have 4 complexes. We have 4 complexes in inner membrane in crystal part. And we have 2 pathways for electron movement. So we have two pathways for electron movement. You see in this picture, this is complex 1, this is complex 3 with orange color, this is complex 4 with green color, and this is complex 2 with yellow color. And we have two pathways of electron movements. So the first pathways begins with complex 1 and begins from NADH. Electron comes from NADH, goes to the complex 1. The complex 1 has prosthetic groups named FMN, has a prosthetic group named FMN, and complex 1 has a FES proteins. Electron goes to the FES protein and then goes to the coenzyme Q, CoQ or coenzyme Q, coenzyme Q, and then go to the complex 3. In complex 3, we have some kind of cytochrome. Cytochrome BL, BH, cytochrome C1, and electron goes through the cytochrome BH, BL, and cytochrome C1, and then goes to the cytochrome C. Cytochrome is a kind of protein similar to hemoglobin. They have a hem group with, for example, iron in the middle or Cu in the middle as a part that give the electron and release the electron. The metal part in the middle of the hem group. Cytochrome is similar to hemoglobin and has a hem group with the iron or Fe or Cu. In cytochrome BL, BH, and cytochrome C1 and cytochrome C, we have a Fe in the hem group. They take the electron and pass the electron through the cytochrome C2, complex 4. In complex 4, we have cytochrome A and A3, A and A3. And in cytochrome A and A3, we have Cu in hem group instead of Fe. And then the electron goes to the oxygen, and we have water as a final product in electron movement change. So H2 water or H2O is a final product of electron movement change. I repeat it again. The first pathways begins from NADH and complex 1. Electrons comes from NADH, goes to the prosthetic group FMN, FES protein, CoQ, CoQ or coenzyme Q, go to the complex 3, cytochrome BL, BH, cytochrome C1, cytochrome C, pass to the complex 4. And complex 4 have a cytochrome A and A3 with Cu in hem group. And then electron goes to the oxygen molecules, and we have a water as a final product. This is the first pathway. In the second pathways, it starts from complex 2. Complex 2, electron comes from succinate, goes to the FAD as a prosthetic group, FES proteins, coenzyme Q, and complex 3. In the first pathways, we have complex 1, 3, 4, and second pathways is complex 2, 3, 4. Complex 1 named NADH, coenzyme Q reductase. Complex 2, succinate coenzyme Q reductase. Complex the coenzyme Q cytochrome C reductase, and complex 4 is cytochrome oxidase. Cytochrome oxidase, NADH coenzyme Q reductase, succinate coenzyme Q reductase, coenzyme Q cytochrome C reductase, and cytochrome oxidase. Okay, do you understand? This part, you understand? Okay. And in this picture, you see the oxysome structure. Oxysome structure or ATP synthesis or F1, F0 ATP synthesis. In oxysome, we have a head group, we have a stock, and we have a base part or base group. In a head group, we have at least 6 subunits, alpha 3 and beta 3, 3 alphas and 3 betas, alpha 3 and beta 3. And also we have delta and epsilon. And in a base part, we have a channel of proton or proton channel or proton half channel. Proton half channel, you see in this picture. When proton transport through this channel, the base part circle around or turn over around self. So we have a conformational change in head group. When the base part turn around itself, we have conformational change in head group. And then ADP join to PI and produce the ATP. So the head group produce ATP. This part of mitochondria produce ATP in mitochondria. We have a head group with at least 6 subunits. This part produce ATP with conformational change because of turn around the base part and proton transport through the channel, and cause of turning around the base part and conformational change of head group made the ATP or produced the ATP. Okay. If you have any question, please ask me. I think it's enough for today. And please someone send me an email. I send you the PowerPoints and nothing else. If you have any question, please ask me.
We don't have a question. This is mine. هاي سعد المايك. تقول لهم تروح تقضي لها قصه وتروح تقضي لها شغله وتقول لهم اذا عندكم سؤال سالوني، وهي تروح تقضي لها شغله وترجع. خلي شويه شاع طباخ. شو هذا فالس؟ هو دائما اشوف يحكي هذا يحكي ايراني بطلاقه. خب اكو عندنا مالتين، هذيك بيحكي فارسي، هو الوحيد اللي مسيطر، اول واحد يجاوب. اقول انا حتى ما يحكي فارسي تبي عايش هناك.
If you have any question, I give your email. I send you the PowerPoint, and I talk about the exam in next session. Do you have question? Okay, thank you very much. See you in next session. Have a good time.
راح مش ممكن. اشوفكم بالمحاضرة.